So now we get to the organismal part. We get to the tour through the different groups of living things. As usual, starting a new chapter, I do that. For some reason, the computer decided to shift its focal point when I did that. And we're going to start with the little guys. Remember, the viruses don't count, so we're going to start with the bacteria. These are picture book suggestions for young bacteriologists. The Diplococci in the hat, Mr. Coccus, the tail of Peter Coccobacillus, and the very hungry Streptobacilli. So the chapter's basically about prokaryotes. The word bacteria is kind of non-specific, but it's pretty much what typically these guys are called. And we're going to work our way, as we often do, through history and germ theory. Is that I mentioned when I talked about microscopes that when you take India ink, which is commercially made, nothing alive in it, and you take a small drop of India ink, put it in a larger drop of water, look at it under the microscope, the particles move. It's called Brownian motion. Now I was taught that it has to do with the vibrations, because everything at a molecular level is moving and bopping and shaking and stuff. And somewhere in the last 10 years ago, I ran across some sort of mention that the old explanation for Brownian motion was no longer completely accepted. And typically for my brain, I went, oh, I should look into that. And it wasn't until I started to write this down that I realized maybe it isn't molecular motion, because I never went and looked that up. Sorry, is that unfortunately, I'm in the frame of mind where, I'll think of something, and if I don't say it immediately or write it down, it flees from my brain within a few minutes. We actually have an arrangement in my family where we're allowed to go rabbit, like the dog in Up, as a way of saying, wait a second, I need to say, even though I'm interrupting, let me say this, or I'm going to forget to say it if I put it off. So as you get older, that may actually be a useful thing to do. But we're going to come up to good old Louis. Louis Pasteur worked with winemaking to a large extent. And when you make wine, the alcohol is generated by microorganisms. When you make beer, it's generated by microorganisms. The beer isn't produced by prokaryotes, it's produced by yeast. But Pasteur, wine, champagne, and that sort of thing, were using bacteria. And so he was kind of tasked with figuring out how that all worked and how it worked well and how it worked badly. Because not only do you depend on prokaryotes to do the job of making the alcohol, if you get the wrong prokaryotes into your mix, you're not going to get what you wanted to get. So he not only was looking at how the bacteria did what they did, but how to deal with contamination. And he was one of the first to kind of realize that some microbes, they didn't differentiate between bacteria and other things at that point. Some microbes needed oxygen in order to thrive, and some of them actually didn't like oxygen, or at the very least could thrive in non-oxygen conditions. So it was the first idea of aerobic versus anaerobic organisms. The idea that some things didn't need oxygen was a fairly new idea. Now, of course, he also eventually worked in other fields, well, all related, but So how do you kill the microbes? How do you sterilize things? Well, certain level of heat they don't particularly like. So there were certain types of chemical treatments that would do the job. And he eventually worked with milk producers because there are bacteria that you can get into raw milk that can be particularly nasty. Interestingly enough, a lot of people exposed to raw milk all the time tend to get along okay with them, but it can be bad for people that are not used to them. They can actually be deadly, which is why you're not supposed to buy raw milk on the commercial market. Pasteurization is a heat treatment. You take the milk, you get it warm enough to kill most of the bacteria, definitely kill the bad ones. If you heat it beyond that level, you will kill all the bacteria, including the ones that will make the milk go sour, but it changes the nature of the milk in a very non-tasty way. So pasteurization had to work out what worked and what didn't work. When you buy milk, there are anaerobic bacteria in the milk that weren't killed by pasteurization. They're more heat resistant than typical bacteria. And they will eventually take the milk sugar, the lactose, and their processes will convert it into one of the alcohols. I don't know if it's ethyl or methyl alcohol. It produces, no it's not, it's lactic acid. They convert it to lactic acid. And the lactic acid will eventually acidify the milk. It'll make the fat blobs drop out, it'll curdle it, it'll change the smell, it'll change the taste. And you can do what's called ultra pasteurization, which was invented like 50 years ago, where you kind of flash heat it. Typically done with a pass-through like a stream, where you flash heat it just long enough to kill those bacteria, but not long enough to change the nature of the milk. But it's expensive, and it's typically done with more expensive things than milk, like heavy cream that can sit on the refrigerator shelves for a really long time before it goes bad. And then there are variations of that they use for milk that they use in the military, where you wind up with milk that doesn't have to be refrigerated, that can sit, as long as it's in a sealed container, it can sit anywhere for a very long period of time and still kind of sort of taste like milk. It does help when you get it into your mouth, it has been cooled, it is refrigerated, because our brains are pretty used to cold milk. But pasteurization. Then we get to Lister. Yes, it is no accident that Listerine has Lister's name in it. He was interested in, as people started to realize that some of these guys were getting into open wounds and causing infections. Lister set his sights on preparation of surgical rooms, operating rooms. And what he wound up doing is treating things with carbolic. Yeah, I wanted to make sure it was carbolic and not carboxylic. Carbolic acid, to the point where when you came into the room, you'd go through a spray of carbolic acid, and there was just sprays all over the place. It turns out carbolic acid is very effective, is that he brought the infection rate on surgeries way down from what it had been. But our own cells are not that crazy about carbolic acid, so it turned out to have long-term effects from people breathing it in and on surfaces and such. So they went to alcohols and other kinds of things eventually. But the idea that you could treat things in a surgical suite to reduce post-surgical infections was very much Lister's idea. He also came up with a very important lab process. They were discovering that with the right sort of nutrient medium, you could grow bacteria, you could grow bacterial colonies in the lab and study them. But it's very difficult taking samples from nature to do that and wind up with just one species of bacterium in your culture. But that's what people wanted. They wanted to be able to study, what particular bacterium is doing this and what bacterium is doing that. And if we have an infection, well, we've figured out the staining and the nutrient preferences of all of these different bacteria. Which ones are we actually finding in these infections over and over and over? Well, to do that, you need to find a way to produce a culture that only has one species of bacterium in it. And he came up with a way of doing that. With progressive dilutions. So you take a sample from your mouth and put it on a plate, and you get a bunch of bacteria. That's not one species, though. So take a small sample and you dilute it. You put it into water or saline solution if you need a saline solution, and you spread it in the water. Then you take a drop of that and put it on another nutrient dish and grow a culture from that. And if you do that enough times, you will eventually separate out one, like say you've got 20 cultures to start. You take your drop of diluted water and you put it on a culture plate, and of the 20, you've managed to snatch 8 of them, and you let that grow. You take that and you dilute it, and you take a drop of that and put it on the thing, and maybe now you've got 5 of them. You do it again, maybe you've got 3 of them. You do it again, maybe you've got 1 of them. Eventually you will get, growing on your medium, just one type of bacterium. How do you know that? Well, it has certain properties. You can look at it under the microscope and see that you don't have a mix to some extent. Microscope's not the best way to tell bacteria apart, but it does help. And once you have a culture that is what they call a monoculture, just one species, then you can just keep perpetuating that and do your research on that. So it's an incredibly important discovery. Then you get Koch, who was trying to decide, okay, so we are finding these different microorganisms when people are sick. Remember, they did eventually decide that the sick people's cells were not transforming into these microorganisms, that the microorganisms were coming somehow into them, or maybe we're already there. It actually turns out that a lot of our microorganisms can turn against us under some conditions. Streptococcus that causes strep throat. Most people have streptococcus in their throat all the time. And some people are susceptible to it growing out of control and your immune system reacting with inflammation, and you wind up with strep throat. Some people never have that issue. But the idea with Koch was, so how do we figure out if this microorganism is really associated with this disease? And so he developed what are called the Koch postulates. Postulates are, kind of an if-then statement. And the postulates basically are say, okay, you've got a microorganism that you think is the problem. Does everybody who's sick with your sickness have it? So you keep sampling and sampling and sampling and sampling, and you are always finding this particular microorganism. Now, unfortunately, like if you were doing strep throat, that wouldn't be the only thing you'd find, but you would find Streptococcus in everybody that had strep throat. You'd find, a handful of other species there as well, because there are other species that we all kind of share in our throats. Can you get it and then culture it using the approach that should work for that particular microorganism, right? Because different microorganisms have different types of nutrient preferences, and sometimes you think you've got something that it turns out to be similar but not exactly the same. And so sometimes you just go, well, we know what Streptococcus will live on in the lab. Let's make sure that this thing that we've got that we're pretty sure is Streptococcus will actually live on the cultures that we've prepared for it. And can we get it from diseased individuals? In this case, can we get it from their throats? That's another thing. A lot of diseases, the bacteria are localized. They produce lesions. And then it's a matter of, can we sample bacteria from those lesions and then get them to culture all the time on the thing we think they are? Can we take bacteria from the cultures and use it to actually cause the disease? And this was back a ways. There are certain were researchers that were actually exposing people to things. But Koch also came up with ways he was one of the early ones that developed animal models. He found out that things like guinea pigs could be used to culture tuberculosis bacteria. And which actually wound up eventually being tricky, because it turns out tuberculosis is actually a combination of factors. But can you take from your culture those purified bacteria and use them to produce the same disease? That's pretty convincing you've got the right thing. That could be, I don't know if that works for strep throat, since we all do harbor streptococcus. I don't know if you took streptococcus from a culture and put it in somebody's throat, whether they would automatically develop inflammation, or whether it would just be like a little party, like, hey, he's a new guy, let's have some fun. I don't know. Keep in mind, I'm a storyteller. I make these examples up on the fly. Sometimes I'm 3/4 of the way through the example and realize I'm not sure this will play out all the way. And then, okay, we've used our cultured microorganisms to produce the disease in another individual. Can we then get those back and culture them the same way? Maybe the thing we're putting in is somehow associated with the disease, but giving it to somebody and the disease happening, maybe they're not generating those bacteria. Or maybe we, along the way, have the wrong bacteria. Culturing and doing transfers and stuff is very prone to contamination. And so on every step, you're just kind of checking and checking and checking and checking. And that's really, Koch undoubtedly was an organizer. Detail, 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 detail. Check, 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 check. And this, of course, is applied to viruses as well. Of course, at that point, you can't, producing virus cultures is very, very difficult to do in the lab. You can't get them to grow on plates unless they're bacterial viruses, and bacterial viruses don't typically cause any kind of issues in humans, just in our bacteria. And viruses that affect our cells can grow in some cell cultures, but they were a ways for being able to take cells out of a person or out of an animal and culture them in the lab. So this wasn't really doable for viruses. It's not doable for every bacterium. A lot of bacteria are very, very picky about what they'll grow on, and you cannot get them to grow on a particular laboratory culture. And sometimes they're just very difficult to tell apart, is that there are a lot of what they call strains. And occasionally you'll get a bacterium, like that was the thing I was saying about strep throat. It doesn't always cause disease, so it might not pass all of these levels. Or in some cases you transfer it, and because of the reaction of the specimen you transferred it to, you get a disease, but it's not the same disease. Diseases that we associate with infections can produce very, very different kinds of symptoms in one individual versus another individual, especially if you've done something and it gets into the blood. So that's the postulates. Still on Coke. That sounded weird. Not too surprisingly, if you're doing this sort of thing, you're trying like crazy to determine what you can get the bacteria to grow on and what you can have in the lab that's fairly stable, that is mixable. You can put different things into it and mix it in and have it be stable. And one of the great things that we do in microbiology is that we have gelatin that itself has nutrients in it. It's the basic material eventually for Jell-O, but Coke developed nutrient gelatin, where you can mix different nutrients into it, and then depending on the nutrient needs of the bacteria, you'll get more growth or less growth or non-growth and some growth, depending on the bacterial medium. Okay, that's a good place to stop, so we will stop there.